So I'd like to thank all the speakers. I'll use David's nomenclature and say it was wicked good. It was a wicked good session. I'll counter with the Oakland way. It was hella good session. We're ready for the panel. So are there any questions for the panelists?
David, I have a
Oh,
question.
okay, yeah.
Lisa Aylward from Summit Toxicology. Thank you all for the really nice presentations. I have a question which is just puzzling me as a person not involved in this field, and that is, how is it when you use your library to do the creation of the mutant pools, how is it that you end up with only one mutation in a given cell, such that you end up with pools of cells, each of which has just one change? I'm not sure I understand that.
Oh, don't push that. That doesn't work. Yeah, I should have made it clear. It's just when you do the lentivirus infection, you do it at a low multiplicity of infection. So you basically are doing it at a concentration of virus such that every cell is going to get, on average, less than 1. Okay. So we usually do an MOI around 0.3. And then what you do then is you select for the cells which have the knockout by using a puromycin, which is just a antibiotic marker. So it's just low infection and then a selection for the ones that have the knockout. I mean, the hope is that each individual cell is only getting one knockout. You don't necessarily know that, and I think that's an important caveat in sort of the interpretation.
So, and then, I'm sorry, the more substantive question perhaps, Dr. Liu, right at the end of your talk, you mentioned that there was some epidemiologic evidence that there were genetic alterations in one of the genes that you had identified in populations that were associated with outcomes related to arsenic. Can you talk just a little bit about that and talk a little bit about how that might extend further, other ways that could be applied?
Yeah, so in this case, that the microRNA 124, because of the time, I didn't get into the biology of this microRNA. It turns out this microRNA is the most abundant protein in the brain. So but it's not specific to the brain. So, but if you sequence the microRNA of human brain or rat mouse brain, and more than a third of microRNA sequences corresponding to microRNA 124. So that's why we sort of, oh, that's interesting. And since my collaborators, they already have this human cohort, particularly looking at the neural development in a cohort of children. So we thought that might be interesting to look at whether there are genetic variants around this the microRNA that may either, I think we don't have any SNPs that alter the targeting, but I think maybe some regulatory SNPs that would regulate the change the expression of that. So basically, David Cresciani and Bob Wright, they have their cohort. They did the actually they already did the GWAS on that. So basically, what we did was essentially pull out the SNPs for that particular region and did the association analysis. Yeah, but I think we were very interested in looking further whether those SNPs actually alter the expression of this. But right now we don't have, you can't get human brain sample. So the only thing you can look at is get the blood sample looking at the PBMCs. But that again, whether that really correlate to what's going on so that's the issue that but yeah.
Hi, Rick Wycheck from NIEHS. So this question for Kwang and for Chris. So how many different cell types did you test your system on? And so are you getting the same hits when you use different cell types, which would imply that the mechanism of the toxicity in different cell types is the same or different?
Yeah, I mean, I think that's a significant limitation of this study. So in this case, we use specifically the K562 cell, which is a hematopoietic, and it's a cancer cell. And so all we can say is that this is true for this cell line. I think it would be really useful, and certainly something that we would like to do, which would be to try a variety of different cell types to see if this is true across cell types, or whether this is a cell type specific. There is one hint, which is there has been some work on red blood cells and arsenic, suggesting that they may have a unique export system for arsenic, and specifically the arsenic selenium complex. So that suggests there may be some red blood cell specific mechanisms that we touched into. And I guess I'd sort of point out that even though we were both working on arsenic, we came up with a very different set of genes. And so I think that sort of points out in maybe a limitation or maybe a strength of the approach is that we are identifying multiple different pathways that are involved. And I think the answer you're going to get is going to depend to a certain extent on how you ask the question. So,
yeah, I think this is a very important question. I mean, we sort of did the screen using the serum model that we already established. Actually, we did the RNA interference screen on using the serum model. But I think your question is very important. I mean, I think that really depend on different cell types will express different genes. So, for example, you may knock out a gene that's not even expressed in the cell line you use, so it's essentially you already eliminate that. So I think that in the end, what we hope for is to do screen in multiple cell lines. Or on the other thing is that once we have these important hits, we validate in different cell types after. So sort of like do the primary screen in one particular cell, that's more labor intensive, expensive. Afterwards, we validate. In fact, the microRNA 124 is lowly expressed in the cells we did the screen on, but it's much higher expressed in the neuronal cells. That right now we're actually doing functional follow-up of that screen result in the neuronal cells. But that, your point is well taken. Yeah.
Yeah, I just think it's really an interesting approach to be thinking. It really really brings to bear a whole new thinking about toxicity. So you've got different cell types in the context of the organism. How are they all responding? So collectively, you're going to have a toxicity effect. And is one of the cell types the predominant that actually leads to the demise or the phenotype in the organism?
Yes.
Very interesting.
Yeah.
Here's Neela Luru from Utsolo Oceanographic. This question is for Dana. So this question is for Dana Dolnay. Are RNA-dependent methylation heritable?
So once the genome region is methylated, it should be heritable from cell to cell. Are you talking about in a multi-generational?
Yeah, multi-generational. Yeah.
It would be subjected, we don't know. I mean, I haven't done that experiment, but I would suspect that it's like all other forms of epigenetic reprogramming, that what we see that escapes that multi-generational reprogramming is relatively rare, but there is evidence that it happens.
Okay, thank you.
Dave Gerhold from NIH NCATS. I wanted to ask Dr. Liu to walk me through the process by which you figured out which gene knockouts were enriched in your final population.
Sorry that I mentioned that I didn't have time to put the bioinformatic process of analyzing the data. So the data analysis was done by Blanca Himes, who was a research associate at the Channing at Harvard Medical School, but now recently she moved to UPenn. But anyway, so there some labs, like in the Blanca's lab, have established the sort of like pipeline to analyze both like high throughput data, both RNA sequencing data, as well as this CRISPR screen data, because essentially so in general, what you do is that after you have these cells, let's say cells that in the collection of population of cells that respond better to arsenic, you have these cells after the screen. Then you have a sort of screened cells, like the control. So you extract the genomic DNA, right? Because these CRISPR guide RNAs are integrated stably into the genome, so they are there. So basically, then you do the PCR because all of them actually contain same flanking sequences. So you can use the same primer, but inside is different. Barcode is different. The actual sequence is different. So see what I mean? But on the outside, they're the same because that's how they were designed. So basically, you sequence your actual sample and your control, right?
I missed that they're stably integrated, and so you're looking.
Stably integrated. So therefore, the genomic DNA you extract from the cells will contain the DNA sequence there. You PCR them out. You PCR them, the control out. Then you sequence, meaning that you just the sequencer will take your barcode and then read through your sequence, right? Just do sequence, high throughput sequencing, and you generate hundreds of millions of sequences. Then you map, in this case, in RNA-seq case, that you map to the genome because it corresponds to any regions of your protein coding gene in messenger RNA. In this case, you already know that your starting material, right? This 20,000 genes with 6 guide RNAs, the sequence is known. So you just match your sequenced stuff to the actual sequence of this thing in the library to begin with, right? Mm-hmm. And then you map. Okay, so for gene A, the guide RNA 1, I have 20 copies in this. In here, I have 100 copies. This normalized, meaning normalized against the total sequence. So therefore, you got the representation of each of the guide RNAs in 2 samples. That's how you got you can divide, right? And you get the enrichment for.
Okay. So would this be sensitive enough that you could see which ones drop out as well?
Yes. Okay. That's the idea. Yes.
Thank you.
Yeah.
I'll just add that the analysis of the data that comes from these whole genome screens is rapidly evolving, and it's kind of similar to the time point we were when we were getting RNA-seq data for the first time, or microarray data, that the approaches and technologies are rapidly evolving, and there are many bioinformatics groups who are coming up with the best way of dealing with the data. But I will say that it is not something that's not approachable by anyone who's dealt with RNA-seq data. It's very similar to that, and the results that you get from it are similar. As I mentioned, the added twist, though, is that you have multiple sgRNAs, and so it's somewhat difficult to make a decision over what's your cutoff? Do you think 1 sgRNA is enough? Is there 2 or 3? Or 6 or 8, and I'm not sure there's a good answer to that, and I think, we took the approach of basically taking a very low threshold and then doing a secondary screen with a much higher threshold, and our thought there was that since we don't know how well these sgRNAs work at this point, then we should be as open as possible. I do think, though, that as validated sgRNAs now are becoming more and more available, where we actually have shown that an sgRNA does function and does work, there are libraries of these that are now being generated, that's going to be less of an issue. And we're going to know that this sgRNA, at least in this cellular context, works. And so therefore we can be more assured that if we see that sgRNA, that it will knock out the gene, and that we can trust that if we don't see it, it's not because the sgRNA doesn't work, it's because the sgRNA actually, that the knockout isn't doing anything that we think is phenotypically interesting. So it may be that in the future we won't need to use 3 sgRNAs. Maybe we'll just have to use the one validated one, which we know works, And that will greatly increase or decrease the scale of these screens, and I think make them much easier and faster to do. Because one of the things is with these screens is that you have so many sgRNAs, you have to use a real bucket load of cells, and that's why we use these cells that are really easy to grow, like K562, because you can just grow tons of them and it doesn't really matter. I think what will be exciting in the future is that if we have these validated systems, then we can use the more difficult cells like a neuronal cell line or something like that, where we really want to ask a question. You can't grow bucket loads of them. I mean, you can, but you'll need, I think, the proverbial 300 undergrads to grow your your population of cells. So.
Yeah.
So, Shruti Mahalingaya, Boston University School of Medicine. I have an applications question. I'm a reproductive endocrinologist, and we often freeze embryos and gametes, or oocytes, in a vitrification process that uses dimethyl sulfoxide as a cryoprotectant. There have been case control studies showing increased bone cancers in children born after cryo procedures, very rare and not enough to change our clinical practice. How could one conceivably use genome editing tools to assess whether DMSO could be a concerning exposure to the germline or embryos? And this is open to any of the panelists. Thanks.
Well, I'm not sure you need to do genome editing there. I think you just want to do sequencing to see if there's any, at least classic genetic changes there. The epigenetics would also be similar kind of a thing. You'd expect to see it if this was a real phenomenon. DMSO does have lots of biologic activity, so it wouldn't be completely surprising.
Yep. Sort of ethics aside, this is, we have tried a few times to get grants funded to look at egg cryopreservation and changes in DNA methylation and chromatin structure, similar to what we know with IVF, that it has these statistically significant but very small increases in these imprinted disease disorders. So I think this is, ethics aside, an area where something like a pi RNA approach could work in theory. So if you had identified regions in the oocyte that underwent genome-wide, or demethylation, if you wanted to protect those regions by, co-incubating the cells with pi RNAs that were specific to that region, you might be able to protect the eggs from that demethylation event. And you can imagine other epigenetic applications for that as well.
Yeah.
Ken Buckelhead, Brown University. So I'm thinking of sort of a screening approach to toxicity testing and how it might be widened and improved. And by analogy, with carcinogenesis, we have oncogenes, we have tumor suppressors. Maybe there are 100 pathways that are involved in all of oncogenesis that explain everything we know about molecular pathways by which cells can become malignant. In toxicity testing, maybe there are a couple hundred toxicity pathways or pathways of toxicity that are the end stage actors of the bad stuff that happens upstream. So I'm wondering how we might be able to use the tools that you're developing to identify initiating events, which may not be actually necessary for toxicity screening, but would be very interesting, pertinent, and informative for us to understand how these agents act. And for most, maybe they're not very selective, so all we get is sort of this end-stage activation of ER stress or oxidant stress. But are there ways in which we can use these tools to get more insight at the very initiating upstream targeted molecules that are involved, if they are actually there? Because we're going to get overwhelmed in signals that are sort of downstream by these kinds of screening approaches, right? Because you're going to get lots of hits down in the downstream activity. Does my question make sense?
Yeah, I mean, it's just, I can only give a very general response. I think that the Tox CRISPR library that Chris lab is making, in Lopin's lab is making, will be very useful to do that, because those are the genes we collectively select from the literature that are known, as you describe, as a toxicology pathway, toxicity pathways. Some of these are secondary, some of these are maybe initiating events, So with this sort of reduced or focused library, you will be able to actually do a screen, identify which are the initiating event, pathway or genes that really determine the toxicity, not just associated with they are just up and down when you treat the cells with certain toxins, So I think that, doing a lot of screening in different cells and using different toxicological outcomes, you may be able to tease out, which ones more specific to one specific type of toxicants, right? And in particular, if you can design, the whole thing is that for genetic screening is that as a person who trained with molecular genetics is as long as you have a defined phenotype, you can do anything almost, right? Sometimes it's not because the screen did not work, it's because your outcome is not well defined, For example, if you just use cell growth as a phenotype, there are so many things can contribute, right? Oxidative stress, ER stress, and almost anything can have a potential impact on how the cells proliferate, and not necessarily the cell cycle genes, but a lot of genes. So I think if you can design sort of assays that can capture these more subtle, toxic, phenotypes, then you will be able to do that. That's my response.
So I just wanted to add to that, and I'm going to dodge the question. So I'm going to say that this approach by itself may not do that, but one of the things that we've been thinking about is combining it with sort of chemoproteomics sort of approaches. And so for the audience who's not familiar, this is sort of looking at the direct interaction between a toxicant and the proteome, and there are methods to do that. But it's sort of in a similar sort of light, the problem is that you get a lot of things that a compound will interact with, and you don't know which ones are important. Well, I think what this approach allows you to do, the CRISPR approach, is it allows you to ask, well, which are the important ones? And so we think that maybe the combination of a chemoproteomic approach, which identifies all the intercellular targets and maybe could give you a list of the potential molecular initiating events, combine that with some sort of CRISPR approach. And in that case, you're asking, well, of those hits, right, which are the ones that are actually important? Which are the ones that if you muck it up, you have a bad stuff happens, or bad stuff doesn't happen, right? And so I think maybe the combination of these sort of two technologies that are going at it from two different directions could help you narrow down to the set of critical genes that are really involved in the molecular initiation, but also then later in the cellular response to that. But I think it is, you pointed out a really important caveat of this, is that we are looking at the array of responses to the cell. And in a way, I think we have to, though, take advantage of the existing information that's already out there on how these proteins interact with each other. And so there are networks of interactions, so-called interaction networks. And maybe what we can do is we can also work backwards from that. We can look at the set of responses that we see and then work towards that hub, that maybe is converging on a particular protein and saying, well, maybe this is the protein you should be looking at because all of the results seem to fan out from there. So I think those sort of two approaches, taking advantage of our existing information infrastructure that we already have, as well as novel technology, could maybe help us identify those individual molecular initiating events.
Good. Thanks.
Hi, Rick Wychick, NIEHS. I've got a question for the 5' end of the table. So Dana, a question, actually I'm fascinated by your system. And so I'm just wondering, do you think it's the 2' methyl that's actually conferring stability of this particular, of the pi RNAs? Or are there going to be other features of the piRNA that you're going to have to factor into the equation to make it stable enough to elicit a biological effect?
Yeah, that's a great question. As Penny and I were reading the materials we provided on piRNA, one of the articles is, focuses on they ignore the transcriptional side of RNAs, focusing more on the RNA degradation. But the one that they did highlight was siRNA. And I said to Penny, why are we doing this with siRNA too? And it really does boil down to that O' methylation and the stability. So we're doing some testing to see how long the piRNAs remain around, and if we can detect that piRNA in the tissues after we collect after we sacrifice the organism. So I don't have a good answer for you, but I think there are other ways to capture some of these specific characteristics of non-coding RNAs, and it's just a little bit of a simpler, maybe it will turn out to be too simplistic technique, but we should have the answer to our in vivo experiment in the next couple of weeks and know whether the yellow mouse became more methylated.
I mean, it's interesting you have this 2' methyl at the 3' end. Is there any evidence that there may be other modified nucleotides that may be contributing to the stability?
I'm not aware of any other evidence, but that's just like we talked about earlier, the absence of evidence. Penny,
Right.
do you have any comment on that?
piRNAs aren't the only small non-coding RNAs that have that modification. There are other SNO RNAs that also have, and other small microRNAs that have the same modifications, but piRNAs, predominantly mature piRNAs, have that 2' methylation mark, which with the current literature, they say that the main function is to make it more stable, but we don't know much about the somatic piRNAs in general, so it is possible to have other modifications in the somatic piRNAs because we don't know anything about the somatic piRNAs. I hope that answers the question.
It does. Thank you.
I have a quick question for Juan, Dr. Liu. So you identified the polycomb repressive complex. So did you see any hits in any long non-coding RNAs? Were you looking at that at all in your screen?
I don't think the library contains long non-coding RNAs. It was not the first library. I think it's the first GECKO library. Maybe Chris can correct me. I believe it's only gene coding, the protein coding genes and microRNAs, not long non-coding RNAs.
So that would seem like a cool thing to do if they could incorporate that
only
into the library.
technical problem with the RNA is that the knockout is most likely is not going to work because you see what I mean? Small indels are not going to change the RNA structure much. Unlike protein, you have a one frame shift, then you can change the protein. But that's why in fact, it's a different thing. I guess which maybe I can say a few words about it. It's people already begin to design. You have to take out the whole RNA to see the effect. So it's different. You have to design two targeting, take the whole thing rather than just chop the small pieces. So, yeah. But that's an interesting question. That might, you would expect some of the long links might be there, but
Yeah. Keith Halke, PA. So back to the idea of cell choice for these full genome type of screens. I hate to think that we're going to spend the next 10 years or more applying this really powerful technology to some what I think are really outdated cell models. And understandably, it is early, and we need to make sure these things work. But what really are the limiting technical features of doing this in, say, primary cells or 3D culture models? I mean, is it just cell proliferation, cell number, or are there other things that really that we should be thinking about now and focusing on, and maybe devoting more resources to up front rather than continuing?
Yeah, so for sure, I mean, I think it depends on your endpoint and your assay. So we're using initially the simplest assay, which is basically does the cell grow? And we're using that as our phenotype indicating that it's responding to the toxicant. And if it doesn't grow, then we say it's toxic. So, so in order to use that endpoint, we need to work with cells that are able to grow and grow relatively robustly. And so I think that's the limitation with primary cells, is just the ability to get sufficient number and for them to proliferate long enough. And the problem is as you know, with primary cells, they are in a slow but steady process of dying. And so you're always challenged to know, are they dying because I've abused them, or are they just dying because they're primary cells? So, but I think there are models, such as you're well familiar with, such as the HEPA-RG or other models, where maybe could be better mimics. I think the challenge with working with things like spheroids or other sorts of models is, again, I'm going to use a line from somebody else, it's a delivery issue, not so much an issue of the technology. If you can get it in, it's going to work. The problem is how do you get it into every cell in a multicellular system? And so it may be possible using things such as AAV, but that could work. But so I would say those are the two challenges, and I absolutely agree. I think doing this in the best cell line, and if we can find a primary cell line that would work. The other approach, which Dr. Liu used, is to come up with a better assay. So if you have an assay that's not so dependent on the proliferation of the cell, you have some other readout of toxicity, whatever that may be for you, then yeah, you could use a better cell line, such as a primary system. And I think I agree with you absolutely that working towards that, it would be a really useful end to actually do it in an appropriate system as early as possible, so we can get biologically relevant results.
Do we have any comments or questions from the web? Nope. So it's actually time for our break, but we could keep, I don't know if people want to keep asking questions or take a 20-minute break. I think we're going to take a break. All right, great. Thanks everyone.